Good morning, everyone. I am Pushpamna Singh, a PMRF scholar from IIT Kanpur, and I am going to take the problem solving session for cell culture technologies, which is an NPTEL course, and its course instructor is Professor Manak Das. So, before I start the session, I want to make 2 3 points clear. So, the first thing is that I'll be taking in this presentation or in this session, I'll be taking the week 0 assignment, the questions from the week 0 assignment of cell culture technologies, and but at the end of the session, if you have some doubts or if you feel that you are not satisfied with some explanation for any question, you can ask those questions at the end of my presentation. And the thing is that even in the middle of the presentation, you can type your questions in the chat box and I can answer from there. This session is of 1 hour duration. However, if you feel that 1 hour is over and your doubts were not answered, then you can have those questions in the chat box. So I will address those questions in the next session. Or what you can do, you can join the next session, and at the start of the next session, you can ask me those questions. Okay, so let us start now. So this is the first question, that is cells that are they show some property or they have some property need to be cultured while attached on a solid semi-solid substrate. So there are 4 options. The first option is living, the second option is non-living, the third option is anchorage dependent, and the fourth option is anchorage independent. Now there is a very strong clue in the question itself that says cells that are cells that show some kind of property need to be cultured while attached on a solid or semi-solid substrate. So anything that is going to be attached to something or some substrate, that that it is going to anchor itself to something. So from here the answer can be cells that are anchorage dependent need to be cultured while attached on a solid semi-solid substrate. So the correct answer here will be C, that is anchorage dependent. Now here for our cell culture basics, what happens is most of the cell lines or most of the cells that we obtain are basically anchorage dependent. Anchorage dependent meaning these cells need a surface, that is the surface of a flask or the surface of a dish to attach to. Here you can see there are some 4 5 cells, and once they multiply they cover the entire surface, and cells which are anchorage dependent form a monolayer on the flask surface or on the dish surface, and once they have formed a monolayer, they do not go on forming multilayer. So this is the normal property of any cell except for that of hematopoietic cells, which are blood cells. So here, as you can see, normal cells will always stay anchored to the surface or to some other cells. For example, you can see in this flask here, there's a growth medium which is clear, as you can see, but at the bottom of the flask where they have found a surface, they're attaching and they're growing. However, for cancer cells, this property of anchorage dependence is not true, because cancer cells lose this property of anchorage dependence because they have to metastasize to new locations. They have to travel to new locations. So for this question, the answer is anchorage dependence. Moving on to the second question. Which of these chemicals is used as a protective agent when cells are stored frozen? So the first option is only DMSO, second option is only glycerol, third option is both A and B, fourth option is none of the above. So here both DMSO and glycerol are used as a cryoprotective agent. So here the correct answer will be both A and B. Now for storing cells, whether it is bacterial cell or whether it is mammalian cell, DMSO and glycerol are used, and they are used so because They prevent the formation of something which is called ice crystals. So you have your vial where uh, which is stored at ultra low temperature, and it has your cells, your media and everything. So here, if they go at very low temperatures, like something between minus 80 to minus 196, if they are stored at this low temperature, then there is a possibility of formation of ice crystals. So DMSO and glycerol are those agents which prevent the formation of these ice crystals. Now what happens when we use, like what is the procedure for using DMSO and glycerol as protective agent? So first, suppose you have your culture dish where you have your cells. So either by using trypsin or by the use of a physical method such as cell scraper, you will remove those cells. So you will collect your cells. Then if you are collecting cells by using trypsin, then you will have to remove trypsin. So trypsin is a protease. If you leave your cells too long in trypsin, then it will degrade your cells. And like the DMSO, the media that we use, DMEM, it has like if it has trypsin, it also has serum. So it is going to interfere with your freezing process. It is going to interfere with your cryoprotectant. So in order to do that, what most of the labs do is that after collecting cells, they centrifuge cells, like they centrifuge that entire mixture having cells, so that they can get like they can get cells at the bottom, and all your residual media, trypsin, etc. will be on the top. So here you will have your cells. So from here you can collect those cells and add your cryomedia. Now this cryomedium will have somewhere around 10 to 15% DMSO, all your glycerol. So this will act as a cryoprotection or cryoprotective agent. So this was a protocol in brief, just for your knowledge. So, but the answer to this question is both A and B. Both DMSO and glycerol are used as cryoprotective agent when cells are stored in frozen form. The next question is also a follow-up from the previous question. Now, cryoprotection of cells is done at temperatures. First option, below 0 degrees Celsius. Second option, below minus 130 degrees Celsius. Third option, above 0 degrees Celsius. Fourth option, above 100 degrees Celsius. So, as I mentioned in the previous slide, cells are stored, the final storage is done at liquid nitrogen, and the temperature of liquid nitrogen is minus 196 degrees Celsius. So here the answer will be B, that is below minus 130 degrees Celsius. Now, an important thing to remember while storing cells in liquid nitrogen is that storage is done in the vapor phase of the liquid nitrogen and not in the liquid phase. So storing at liquid phase may harm your cells, and that is why like if you have a liquid nitrogen tank where it is half filled, suppose it is not half filled, it is one filled to one third level, the liquid nitrogen is still one third level of the tank, then it is always prudent to store your cells at the vapor phase and not directly immersed in the liquid. Now like since we are talking about cryopreservation at different temperatures, so here what happens is, just one second. Yes. So here what happens is most of the laboratories and one way which is also the ideal way to work is that you do not directly like keep your cell in the cryomedia, and you do not directly keep them in liquid nitrogen. So especially for cells which are very sensitive, this method is not good. So what is done is that first when you have your cells in cryomedium, then you go on gradually decreasing the temperature. So first from 37 degrees Celsius, the optimal temperature at which your cells are cultured. From there, when you add cryomedium, you keep the cells at 4 degrees Celsius for some hours. Then you keep the cells at minus 20 degrees Celsius for some hours. And then you move on to minus 80 degrees Celsius for overnight or for some 24 hours. And then you go to the liquid nitrogen phase. So, especially for finicky or for sensitive cells, this is the way which is preferred. However, most laboratories, what they do is that they start from this stage, that is, they keep the cells at minus 80 degrees Celsius for like 16 to 24 hours. And then after that, they transfer the cells to the liquid nitrogen tank, like in the vapor phase of the liquid nitrogen tank. Now, moving on to the next question. So, the next question is aseptic techniques are required A. To prevent contamination in cell cultures. B. To ensure contamination in cell cultures. C. To ensure non-sterility of cell cultures. And D. To prevent only fungal attack. So here aseptic techniques will be required to prevent contamination in cell cultures. Now, it is very important to prevent contamination in your cultures, and that is because if your cells, so the media that we use uh, in cell cultures is DMEM, plus we also use serum. So, this is a highly enriched media. In order for the best growth of your cultures. Now, since this is an enriched medium, what will happen is like if your if bacterial cells if bacterial cells or fungal spores are like if they get into this medium, then what will happen is they will have a lot of nutrients on which they can grow. So it is very easy to get contamination in cell cultures, and hence it is very important to maintain aseptic techniques like washing your hands before you go on touching your culture plate, and wearing gloves, and not wearing the same gloves again and again, not taking the gloves out once your work is done, like out of the cell culture room once your work is done, having UV in your hood, having even UV in the entire room. So these are some of the basic aseptic techniques which are followed, along with some personal like some personal ways in which to avoid contamination in cell cultures, because once the contamination comes, your entire your entire plate, your entire dish, your entire flask will be wiped out in just in a matter of 1 day or 2 day. So not only bacteria and fungus, sometimes mycoplasma contamination is also seen, and this is the worst contamination to have in your cultures, because mycoplasmas are not going to just be restricted to just one plate. They will be like in your entire incubator, in all the plates, in all the cell lines which are in your incubator. So it is always a good thing to maintain good practices while you are in the cell culture lab. The next question is, cells prefer to attach on, first option, hydrophobic surfaces, second option, hydrophilic surfaces, third option, fluorinated surfaces, fourth option, all of the above. So here cells will definitely not prefer to attach to hydrophobic surface, that is water repelling. So cells will prefer to attach to hydrophilic surfaces. Next question is, which of the following molecules used for surface coating for cell attachment is of organic origin? So again there are four options. The first is aminosilane, second is poly-L-ornithine, third is poly-D-lysine, and the fourth is collagen. So before I discuss about the answer of this question, there's one thing that I'd like to mention. So here, most of the uh, like the widely used molecule that is used for surface coating for cell attachment is poly-D-lysine. So it is basically polylysine, and there are two forms, poly-L-lysine and poly-D-lysine. Any of the two forms can be used. Now, what polylysine does is it provides a positively charged surface, like the bottom of the dish is coated with polylysine. So, most of the coated plates that we get actually have a polylysine coating. And what it does is that it provides a positively charged surface for your cells to attach to using their negatively charged surface at a cell membrane. So this helps a cell adhesion, or so that it can attach to the surface. However, polylysine is a synthetic molecule. It is not of organic origin. So here, other chemicals of organic origin are also used, and from all these options, the correct option is collagen. Another chemical which is used for surface coating for cell attachment is fibronectin. So the next question is, cell culture media by the name of DMEM stands for. So this DMEM stands for Delbecque's modified eagle medium. It is an enriched medium which has bicarbonate ions for a pH buffering system, and it does not contain FBS or the fetal bovine serum. However, it has bicarbonates, glutamine. So we add like something around 10% FBS to make it enriched, and this is the medium Delbecque's modified eagle medium. It was named after the scientist. Next question is, phenol red indicator is primarily used in cell culture medium for. So here phenol red indicator is used for pH indication. So phenol red has a wide variety of like colors which it can show at different pH. So This thing will also come in a later question, but I'll just like to show you. Yeah, I'll just like to show you what are the different colors that phenol red can show. So here, if you look here at pH 6.5, it is like yellowish in color. At pH 7, it is somewhat orangish in color. At pH 7.4, uh, it's like this. And at pH 7.8 or at like basic pH, it turns pink, or at more basic pH, it turns purple. So, like at acidic pH, it becomes yellow, and at highly basic pH, it becomes purple. So, like these are the range of colors which phenol red can show, and hence, in order for pH indication, phenol red is a very preferred indicator which is used in cell culture practices. So our DMEM is mostly it has phenol red, but it like there are some variants which are phenol red free also. Next question: which of these cell culture media is carbon dioxide independent? So here DMEM is CO2 dependent medium. That is because it relies on that bicarbonate buffering system, which I'll explain in a later slide. So it relies on the bicarbonate buffering system, which requires 5 to 10% of CO2. However, out of all these, this L15 medium is a CO2 independent medium. Now the next question is, fetal bovine serum or FBS is extracted from fetal calves. Which of the following statement is true about FBS? So the four options are: it contains cells, it contains fibrin, it contains clotting factors, it contains growth factors. So FBS here contains growth factor. It is a highly enriched serum, and it aids in cell growth. So the questions that I discussed till here were from the week 0 assignment. Now I have some additional questions which also pertain, which are similar to the questions that we discussed earlier, but which will add up to your knowledge of basic cell culture technology. So the first is, what exactly is a cell line? Is it a multi-layer culture? Is it a culture of transformed cells? Is it multiple growth of cells, or is it subculturing of primary culture? So a cell line is basically, so you have your cells. Suppose you have one organism from where you got these cells, and now, so this is something which is called a primary culture. Primary culture, as the name suggests, you are getting the cells directly from the source. So you obtain the cells from your source, which is the primary culture. Now in the lab, you are continuously like culturing those cells. So from here, your cells will divide more. From here, these cells will further divide. Then these will further divide. So a cell culture is that you are maintaining a culture which is derived from the primary culture, and that is your secondary culture. And this is what which forms your cell line. So, and this continuously using some cells to start a new cell line, or sorry, to start a new culture is referred to as subculture or passage. So that is how we like refer to the culture. So if this is the primary culture, then from here we'll start our P1, that is the subculture 1, then P2, then P3, and so on. So this is your passage. So a cell line is a subculturing of your primary culture. So moving on to the next question, which is name the type of culture which is prepared by inoculating directly from the tissue of an organism to culture media. So here we have to name the type of culture like which is prepared directly from the source or from the organism. So the four options are primary cell culture, secondary cell culture, cell lines, or transformed cell culture. So as I discussed in the previous question, here the type of culture which is prepared by obtaining directly from the source or directly from the tissue of an organism will be our primary cell culture. Now the next question. In the secondary culture, cells are obtained from, again as I discussed in the first question, the cells will be obtained from the primary culture when we are talking about secondary culture. The cells will be obtained from the organism when we are talking about the primary culture. But for secondary culture, we get the cells from the primary culture itself. Now the next question. Under favorable conditions, the protoplast can grow into a, now here, it can grow into a callus culture, can go into an organ culture, a root culture, or a shoot culture. So protoplast is your are your cells which do not have uh, like which do not have your protoplasm. So here the protoplast will grow into an undifferentiated mass of your callus culture. Next question is, which of the following is not the part of growth medium for animal culture? So here the four options are starch, serum, carbon source, or inorganic salts. So inorganic salts are a part of your growth medium in order to maintain the osmotic balance. Carbon source is a part of the growth medium for a pH buffering system. Like, sorry, CO2 will be part, CO2 and bicarbonate will be a part of pH buffering system. Carbon source will be required for providing the basic source of nutrients, basic source, for example, glucose, etc., for cells to grow. Serum is to provide the important amino acids. However, starch, which is a complex carbon source, is not a part of growth medium for animal culture. So here the correct answer will be starch. Next question is, hematopoietic cells are an example of, so hematopoietic cells means your blood cells. So as we know, blood cells travel through your blood vessels and they are not adherent. So here there are four options: suspension cells, adherent cells, stem cells, and living cells. So here your blood cells are not adherent, they're not stem cells, living cells. So hematopoietic cells or blood cells are an example of, sorry, yeah, of suspension cells. The next question uh, is the growth of tissue explants in an artificial medium. So here there are different options: tissue culture, primary cell culture, horticulture, or cell culture. So here the answer will be cell culture. Now the next question is the method of maintaining a whole embryo or organ excised from the host organism in an artificial medium. So this is a method of maintaining a whole embryo or organ which is excised from the host organism in an artificial medium. So the option can be organ culture, explant culture, horticulture, or cell culture. So here you see it is a method of maintaining a whole embryo or an organ. So here the answer will be organ culture. Now, virus cultures require the cell cultures of different living cells of mammalian, plant, or microbial origin as host and support for the growth and something of the virus. So, shelter, replication, propagation, proliferation. So, viruses require host for growth and for replication. Without a proper host machinery, viruses, if host machinery is not there, then viruses will not be able to replicate and hence they will not be able to multiply. So viruses require host and their molecular machinery in order to replicate. Now, vaccines for measles, rubella, etc. are currently made in cell cultures. That is true. So these are a replacement for extracellular matrix in artificial cell culture media. So there are four options: hydrogels, meshes, bones, muscles. So here the correct answer is hydrogels, and hydrogels are new and they have been used as replacement for the extracellular matrix in cell culture. Now, cell culture technique in two dimensions was first discovered by Wilhelm Rau, Alexander Fleming, Francis Crick, or Carey Mullis. So the correct option is Wilhelm Rau. Now, the following are a list of essential components of cell culture medium. Match them to requirements for effective cell culture. So here there are four options: phenol red, glutamine, inorganic salts, bicarbonate. And the four options are here. So a pH buffer will be done by bicarbonate, as I explained in the previous slide. So here bicarbonate will be used for a pH buffering system. Regulation of osmotic pressure and membrane potential will be done by inorganic salts. pH indicator will be phenol red. As I showed, phenol red can exhibit a range of colors at different pH depending on whether the medium is acidic or basic. Now, glutamine will provide glucose and amino acids for respiration. So these are the essential components of cell culture medium, and these are the components which DMEM has, which is the most widely used cell culture medium. So these are the essential components which DMEM has, and that is why it is used as an effective medium for propagation or for culture of cells. Now the next question is, why is CO2 often used in an incubator to culture mammalian cells? So again there are four options: to control cell growth, to control oxygen consumption, to control the pH, or to control or prevent viral or bacterial contamination. So here CO2 is used in order to control pH. So I'll explain the CO2 or the bicarbonate buffering system now. So here what happens is your incubator will have somewhat 5 to 10% CO2, and the medium that you have provided to your cells will have your bicarbonate. So what will happen is CO2 can diffuse in and out of your medium. So once your CO2 combines with water, what will happen is it can find carbonic acid, and which can further dissociate into H plus ions or bicarbonate ions. Now here Le-Chatelier's principle will be followed uh, for this buffering system. So here what Le-Chatelier principle says is that if this reaction can go either way, what will happen is if your H plus ion increases, then what will happen? It will go backwards and it will again form carbonic acid. However, if your H plus ion decreases, it will go forward and it will form extra H plus ion, and this is how this entire buffering system will take place. So in the medium, sodium bicarbonate is present as the reserve bicarbonate. So this is also the buffering system which is present in blood, and this bicarbonate system follows the Le-Chatelier's principle. And this is the different color or the range of colors that that phenol red can show, and that is why it is used as an indicator. So here at this point, before we end, there is a very easy numerical that I'd like all of you to solve, and for that, like, I'll give you 5 minutes, and after 5 minutes, if you have the answer, you can write it in the chat box. If you do not have the answer, then also you can write it, mention it in the chat box, and I'll explain the answer to this question. So let's discuss it now. The question says it's Friday and you are splitting your stock cultures. On Monday, you will need at least 9 million cells ready to plate for your experiments. You also want to have spare to continue your culture for the following week. When they are at optical densities, that is when your cells are at optical densities, your cells double approximately every 24 hours. So the doubling time of your cells is approximately 24 hours. A confluent flask can hold 3 million cells, but your cells will only divide efficiently if they are between 5% and 80% confluent. How should you plate up your cells? So here the important thing to know is that is confluency. So confluency is your cells will divide efficiently only when they are 5% to 80% confluent. Now, an important thing to remember here is that if they are only 5% confluent or if they are even just 30% confluent, that is not the optimal confluency, and in such a scenario they will take time to divide, and at that time even when the doubling time will be 24 hours, your cells will not reach the required density. So it's always better to have 80% confluency but not higher than 80%. If your cells are 90 to 95% confluent, then what will happen is you'll feel that they will reach confluency faster and you can plate more cells. But what will happen is if they are 90 to 95% confluent, then like because of contact inhibition or competition for nutrients, you will see a high percentage of cell death. So you will have more dead cells as compared to your healthy cells. So it is always prudent to maintain confluency somewhere around 80%. Now, on Monday, you need at least 9 million cells ready. And the day that you are working on, that is today, it's Friday. So, in order to have 9 million cells ready, what you can do is that one flask can hold 3 million cells. So, let's assume that we have a flask uh, which is confluent and it has 3 million cells. So like 80% of 3 million will be somewhere around 2.4 million cells. But how much do we need? We need 9 million cells. So 9 by 2.4 will be somewhere around 4 flasks. However, what you need to do is that you need to split on Friday. So if you so that you can get your 9 million on Monday. So what will happen if you will back calculate, you'll find that like So if you played something on Sunday and the doubling time is given as 24 hours. So if you played 2.4 million on Sunday, then on Monday you will have double the amount of cells, like into 2. If you then you will have to calculate how much you need to play on Saturday to reach this, and how much you need to play on Friday again, so that from here it will be this much on Saturday, and from here it will be this much on Sunday, and from here it will be this much on Monday, such that you get your 9 million cells. And along with that, you also need, so here, like, you will have to start 4 flasks in order to get your 9 million cells, but along with that you also need one more spare flask, so 4 plus 1 flask. So I'll leave this calculation to you. If you have any doubt with this calculation, like, it's very simple, you just have to back calculate. However, if you have any doubt with that this calculation, you can let me know in the chat box, or we can discuss it right now. But like, if we add the number of flasks, then it will be 5 flasks. Now there are 2 options with 5 flasks. So here this is too much of cells. So we'll have 5 flasks with this much cell density per flask, such that we plate this 5 flasks with this much cells on Friday, so that we get this much of cells on Monday. So this D is the correct answer. People who answered D, that is the correct answer. If anyone has any doubt again in this calculation, you can unmute yourself and ask me. Okay, so that is all for that is all of the questions that we discussed today. If you have any doubt, you can join the next session. And since we are out of time, whatever questions are in the chat box, I'll address them in the next session. And if you have any doubt, do join the next session. Or if after going back, if you feel that any question you could not understand any question properly, you can again ask it in the next session. So do join the next session. And for now, thank you very much for attending the session.